So endomembrane system, these are a group of cell organelles which are distinct, even though they are distinct in terms of structure and function. Some of the cell organelles, they have coordinated functions. That means they function together in a systematic manner. Such cell organelles form a system, and that is called endomembrane system. So this endomembrane system is mainly composed of endoplasmic reticulum, Golgi complex, lysosome, and vacuole. So this is nucleus. So just next to the nucleus, you'll find endoplasmic reticulum attached to it, like this. And close to the endoplasmic reticulum, you might find Golgi body, like this. And somewhere here, you'll find some lysosomes and some vacuoles also. So whatever is synthesized inside the nucleus as well as inside the endoplasmic reticulum, that is mostly proteins and lipids, that will be transported to Golgi body. Now this Golgi body will modify and package all these substances and send it outside to the plasma membrane, either in the form of lysosome or some of these substances will be stored inside the vacuole. So you can see here, it is the functions are happening in a systematic manner. Their functions are coordinated. So this group of cell organelles whose functions are coordinated, they form a system called endomembrane system. So you should remember which are the cell organelles involved in the system. First one is endoplasmic reticulum, second one Golgi complex, third one lysosome, and fourth one vacuole. Now if you consider mitochondria, chloroplast, or any other organelle, they cannot be considered as a part of endomembrane system because their functions are not coordinated with Golgi ER or lysosomes. They have completely different functions. The nuclear membrane could be also considered as a part of the endomembrane system. Okay, so endomembrane system is a group of cell organelles whose functions are coordinated and they work in a systematic manner. ER, Golgi, lysosomes, and vacuoles, they are a part of endomembrane system. So in today's class, we'll be discussing in detail about the structure of these cell organelles. So let's start with the first one. The first one is ER or endoplasmic reticulum. Any idea who discovered ER? Who discovered endoplasmic reticulum? So endoplasmic reticulum was discovered by two scientists, Porter and Thomson, in 1945. So endoplasmic reticulum, you have already studied in lower classes. It is of two types, rough ER and smooth ER, RER and SER. And this is mostly a network or reticulum. The word reticulum means network. So it is a reticulum of tiny tubular structures scattered in the cytoplasm, but usually attached to the nuclear membrane. And also this ER actually has two parts. The inner part of the ER is referred to as lumen. So whatever comes inside the lumen, we call this compartment as luminal compartment. And whatever comes outside the ER, that is the cytoplasmic region, that is called extraluminal compartment. So basically, ER divides the intracellular space into two, luminal part and extraluminal part. Luminal part is the inside of ER, extraluminal is outside of ER. Also, you know that ER is made up of tubular structures, membranous tubules, and sac-like structure. These tubular and sac-like structures are called cisternae. So ER is made up of tubular and sac-like membranous structures called cisternae. So like I mentioned, ER is of two types, rough ER and smooth ER. So can someone tell me why is it called rough and smooth?
Rough because ribosomes are attached on it.
Yeah. Both of you are right. It is called rough because ribosomes are attached to it. So when you look it under the microscope, it appears rough. But smooth here, it does not have any ribosomes attached to it. Because of absence of ribosomes, they appear smooth under the microscope, and that is why it is called smooth endoplasmic reticulum. So we'll be discussing the functions of each of these. Since rough endoplasmic reticulum have ribosomes attached to it, it is mainly involved in protein synthesis. You know that ribosomes, they are protein factories, and since ribosomes are found attached to RER, their main function is to synthesize proteins. So RER is frequently observed in cells actively involved in protein synthesis and secretion. This sentence is very important. Also, you know that this rough endoplasmic reticulum is continuous with the outer membrane of the nucleus. It is attached to the nuclear envelope. Smooth endoplasmic reticulum has more functions compared to rough endoplasmic reticulum. Since they don't have any proteins, since they don't have any ribosomes, they won't synthesize proteins. Instead, they will synthesize lipids. So RER will synthesize proteins, SCR will synthesize lipids. Also, some of the steroidal hormones like testosterone, estrogen. So some of these steroidal hormones are also produced or synthesized inside SCR. Another important function of SCR is detoxification of drugs and poison. So when you consume a lot of unwanted poisonous or toxic substances, for example, alcohol or drugs, so that is why you're not supposed to consume drugs beyond the prescribed dose. So you might have seen on the backside of paracetamol, it will be written you're not supposed to consume a lot of paracetamol because it will damage your liver. Okay, so SCR will be involved in detoxification. It will try to remove all the poisonous substances like drugs and alcohol. So this mainly happens in your liver cells. So liver cells of vertebrates, they have SCR that helps in detoxification of drugs and poison to make it less poisonous. Also in our muscle cells, you know that if your muscles have to contract and relax, you need calcium ions. And these calcium ions are also stored inside smooth ER. So our muscle cells have smooth ER which stores calcium ions, and these calcium ions will help you to contract and relax your muscles. So this type of smooth ER found in muscle is called sarcoplasmic reticulum. So sarcoplasmic reticulum is a type of smooth ER which can store calcium ion required for movement of your body parts. Okay. So we'll do a quick summary. RER has only one function, mainly one function, protein synthesis and secretion. It is attached to the outer surface of the nuclear membrane. Smooth ER has around 4 functions: to synthesize lipids, then to synthesize steroidal hormones, to detoxify drugs and poisons, and also to store calcium ions in your muscle cells. Now, one more point, membrane biogenesis. So this is something you have studied in 9th standard. Do you remember what is membrane biogenesis? What is the role of ER in membrane biogenesis? What do you think is membrane biogenesis?
Transfer of materials through the membrane.
Yes, somewhat, like almost correct. Now, what do you mean by the word genesis?
Genetical material.
No. The word genesis means to produce, production. So membrane biogenesis means basically the production of synthesis of plasma membrane. We have already discussed in the last class that plasma membrane is made up of lipids and proteins. So these lipids and proteins are required for the synthesis of lipids and proteins, which are required for the synthesis of plasma membrane, should come from somewhere. And it is actually coming from RER and SCR. So RER will provide the proteins, SCR will provide the lipids. So basically endoplasmic reticulum helps in the synthesis or production or construction of the plasma membrane. And that is why we use the term membrane biogenesis. So membrane biogenesis is the process by which plasma membrane is synthesized using lipids and proteins produced by endoplasmic reticulum. Okay, so this is how it looks like. You can see here, it is attached to the nuclear envelope. Mostly rough ER will be attached to nuclear envelope. Also, you can see a smooth ER. It does not have any ribosomes. Rough ER, it has lots of ribosomes attached to it. You can also see the lumen. Lumen is the inner side, inner empty space of endoplasmic reticulum. You can also see stack-like or tubular structures here, flat structures. This is called cisternae or cisterns. So next one is Golgi body or Golgi apparatus. So Golgi bodies were first discovered or observed by Camillo Golgi. And that is why the structure was named actually after him, Camillo Golgi. So he found some densely stained structures, reticular structures, that is network-like structures near the nucleus. And later these structures were named after the same scientist, Camillo Golgi. So Golgi bodies are also made up of structures similar to what an endoplasmic reticulum is made up of. It also has a lot of flat structures, disc-shaped sacs or cisterns, a network of cisterns. But these are stacked parallel to each other. So it might look like this, a stack of like cups or bowls, somewhat like this. So this is how Golgi bodies look like. They're stacked parallelly to each other in the form of sac or cistern. Now they usually have two parts. One of the part is called forming phase and one of the part is called maturing phase. So usually the forming phase is the one which is near the nucleus and the one away from the nucleus is called maturing phase. So forming phase is also referred to as cis part and the other one is called trans. So cis is like the receiving end, trans is the shipping end, sending end. Okay. So cis part will receive stuff from ER and nucleus, and the trans part, after modification, it will send substances to other parts of the cell. So the term cis means on the same side. The term trans means on the opposite side. So same side as in it is on the same side of ER and nucleus. It is usually located near ER. And the part of Golgi which is located away from ER and nucleus, it is called trans. So cis region will receive substances from ER and nucleus. Trans region will send substances after modification. Also, I've written one word here, dictyosomes. Any idea what are dictyosomes? Dictyosomes. So in case of plant cells, the Golgi body is actually arranged in the form of isolated units. They are not arranged parallelly or in the form of a stack. They are usually isolated units. So that is why we don't use the term Golgi body in plants. We usually use the term dictyosomes. So they basically have a Golgi body, but we use the term dictyosome because it is not arranged in the form of stack. These are mostly in the form of isolated units scattered here and there in the cell. Okay. Then functions of Golgi, which we have already discussed in the first class, it is mainly involved in packaging of substances. So whatever substances are produced by ER and nucleus, it will be sent to the Golgi body in the form of vesicles, and this will be received by the cis phase of the Golgi apparatus. And inside the Golgi apparatus, this will be kind of modified and packaged. And after that, this will move towards the trans phase, which is the maturing phase. And that is the reason why Golgi body is always found close to endoplasmic reticulum. So almost all the proteins and lipids produced by ER will be modified inside Golgi body. And that is why Golgi body is considered as an important site of formation of glycoproteins and glycolipids. So glycosylation is a process by which sugars are added to proteins or lipids. So Golgi body will modify proteins and lipids by adding sugars to it. and this process is called glycosylation. So Golgi apparatus is the main site of glycosylation. So basically, we can compare Golgi to a warehouse where you can receive, sort, ship, and even manufacture some of the substances. So Golgi body is like a courier office or a post office where packages will be received, then they'll modify it, pack it properly, and then send it to the correct address. So exactly the same thing happens in Golgi body. Whatever is produced by ER and nucleus will be received by Golgi. All these proteins and lipids will be modified by adding sugars or something else, phosphate group maybe. And after that, this will be packaged in the form of some vesicles. Vesicles are round structures. Then they'll be sent to the destination, usually plasma membrane or any other part of the cell. So this is how the Golgi body looks like. So you can see there are two parts here. The upper part is called the cis phase because it is close to the ER. And the lower part is called the trans phase or the shipping phase, which is away from the ER. And this is where, after packaging, substances will move out of the
Sir,
Golgi body. Yes.
sir, screen is not seen clearly.
Please, it's not clear. Screen is not clear. What about
Yes.
others?
Yes, sir. It's appearing blurred.
Okay, I'm not sure. I think it could be because of the picture also.
So not only this one, the previous ones also.
Okay. Could be because of the connection. So give me 1 minute. Let me connect to another network.
Yes.
Is it clear now?
No, sir.
So no.
It has become more blurred.
Okay, then I'll connect it back to the old network. What about now? Is it better?
Same.
Same. Okay, I'm not sure. Like it could be because of the network. Okay, at least you can see the shape, right, of the Golgi body.
So, yes, so now we can see.
So now we can see it's clear.
Okay, cool. Okay, so now it is clear. So you can see there is one side which is close to the nucleus, that is the upper portion. This is called the cis phase. So the word cis means same. So this portion is called the cis phase, close to the ER and nucleus, and the other opposite phase is called trans phase. So cis phase is called the receiving phase, and trans phase is called the shipping phase. So substances will be received in the cis phase, and substances will be sent off from the trans phase. Also, you can see some round structures here. What are these round structures? This one, for example. What are these round structures?
Vesicles.
Yeah. So these are vesicles, and you can see some dots, purple colored dots inside this. So that could be a protein or lipid. So after modification, the Golgi is trying to package it and send it to somewhere else. So it will send these in the form of vesicles. So vesicles are spherical structures, membranous structure, which buds off from the Golgi body and then go to the destination. Suppose if the destination is outside the cell, then this will go and fuse with the plasma membrane. So plasma membrane will send it to the exterior of the cell. So that's all about Golgi body. So Golgi bodies were first observed by Camillo Golgi. Then Golgi bodies are also made up of sac-like tubular structures called cisternae. They have two parts. The part which is close to the nucleus and ER is called cis phase. The one which is away from ER and nucleus is called trans phase. Golgi is mainly involved in packaging and modification of substances. So it will help in glycosylation, that is addition of sugars to proteins and lipids. Is this clear, this slide?
Yes, sir.
Yes, sir. It's clear.
Yeah. Not the colored region. So that is a screenshot from your NCRT. It might appear blurred because it is taken from your book. At least the other things are clear, right? Whatever I've typed.
Yes, sir. It's clear.
Okay. So lysosomes. What is the other name of lysosome? You have studied earlier, right? Lysosomes are also known as,
Suicide bags.
yeah, suicide bags of the cell. So they are called suicide bags of the cell because they contain some strong digestive enzymes. So whenever a cell is damaged, the lysosomes will break open and release these enzymes and kill the cell, destroy the cell. So that is why it is called suicide bags of the cell. Lysosomes are first reported by a scientist named Christian Dider. And lysosomes are membrane-bound vesicular structures. So you should always remember which one is membrane-bound, which one is non-membrane-bound. Then lysosomes are formed from Golgi body. So that is a very important point. Lysosomes are formed by the process of packaging in Golgi body. So in the last slide, we saw some vesicles here. So one of these vesicles will contain hydrolytic enzymes, and that vesicle will be actually lysosome. So lysosomes are actually derived from Golgi bodies. But the enzymes of lysosomes, they are made by rough ER. Okay, so the hydrolytic enzymes are made by rough ER, but it is packaged into a vesicle by Golgi body. So if the question is, lysosomes are formed from which cell organelle, then you should write Golgi body. But if the question specifically says the enzymes of lysosomes are made in which cell organelle, then it should be rough endoplasmic reticulum. So like I mentioned earlier, lysosomes are rich in enzymes called hydrolytic enzymes, hydrolases. So there are so many types of hydrolases: lipases, proteases, and carbohydrases. You have studied some of these enzymes last year in digestion. So lipase, what does it digest?
So lipids.
Yeah. Lipases will
Lipids
digest.
are fatty acids and glycerol.
Yes. So when lipases digest fat, you will get fatty acid and glycerol. What about proteases? Proteases will digest proteins. Carbohydrates, like you have studied salivary amylase last year. So that is an
Right.
example of carbohydrates. So
Tomatoes.
carbohydrates will convert starch to uh, maltose.
Tomatoes.
Yeah. So breakdown of carbohydrates will be done by carbohydrates. So all these are example of hydrolases. So hydrolases are enzymes which can break down complex organic molecules into simple molecules. Now, another important question is, if lysosome contains these very strong enzymes, then why do you think lysosomes will kill the cell only when the cell is damaged? Why not at other times? Why can't lysosomes destroy the cell at other times? So that is because these enzymes are usually inactive. So these enzymes will become active only when the pH is acidic. So usually the pH of the cytoplasm or the a cytosol, it is neutral pH, it is not acidic. And as a result, these enzymes are inactive. But when the cell is damaged, what happens is the pH will change, it becomes more acidic. And when the pH becomes more acidic, it will activate these enzymes, lysosomes will break and destroy the cell. So that is how it functions as a digestive compartment. Okay, so that is the main function of lysosome. So when a cell is worn out or cell organelle is damaged, the enzymes of the lysosomes will become active due to the acidic pH, and it will destroy the cell. Now, lysosome has another function, that is phagocytosis. This you have already studied in 9th standard about how amoeba obtains its food. So amoeba will use pseudopodia, and amoeba will kind of take it inside, and this food item will be broken down with the help of lysosome. So lysosomes will be fused with the food vacuole, and the digestive enzymes inside the lysosome will digest the food. So this process is called phagocytosis, also known as endocytosis. So phagocytosis is actually an example of endocytosis. And we have discussed in the last class, endocytosis is an example of active transport. It requires energy. A similar process is also shown by our white blood cells, that is WBCs. So one such type of WBC is called macrophage. So macrophages are those WBCs which can kill or eat bacteria. So suppose you have an infection or a wound in your body. After some time, you will see that that particular body part will swell due to inflammation. So this happens because WBCs will reach the site of infection and they will kill or eat all the bacteria, engulf the bacteria. So this process of engulfing smaller organisms or food particles is called phagocytosis. And in this process, digestion of these food particles or organisms happen because of fusion with the lysosomes. So this is the first function of lysosome. Second function we already discussed, that is self-destruction. That is, when a cell is damaged, a cell organelle is worn out, lysosomes enzymes will become active and it can kill itself. This is called autophagy. The word auto means self, phagy means to eat. Okay, so when lysosomes destroy itself, that is called autophagy, to recycle the cell's own material. So in 2016, the Nobel Prize in Physiology or Medicine was awarded to a scientist named Yoshinoro Oshumi for his discoveries of the mechanism of autophagy. Okay, so what you see on the screen right now, this is autophagy. You can see here. So there is a cell or a cell containing or a vesicle containing some damaged cell organelles like mitochondria, peroxisome, etc. So lysosome will fuse with this vesicle, and the lysosomal enzymes, hydrolytic enzyme, will digest all the organelle components. This is called autophagy. So if a question is asked in the exam, which cell organelle is involved in autophagy, then you should write lysosome. The name of the enzyme is hydrolases or hydrolytic enzymes. These are active only at acidic pH. Last part of the endomembrane system is vacuole. So vacuole is also a membrane-bound structure. This is also membrane-bound structure. It is mainly like a storehouse. So it will store a lot of substances like water, sap, excretory products, and some other materials which are not useful for the cell. A very important point is vacuole is bound by a single membrane, and the single membrane is called tonoplast. So tonoplast is the name of the membrane surrounding the vacuoles. Also, we have seen earlier that plants have a large central vacuole. Because they have a large central vacuole, up to 70 to 90% of the cell volume will be occupied by vacuoles. All the other cell contents will be pushed towards one side. The type of vacuole which is found in plants is called sap vacuole. So it usually contains some water, some nutrients, and other materials. So this is called sap vacuoles, which could also provide turgidity to the plant. Okay, so turgidity means when a plant is put in hypotonic solution or pure water, the plant cell will absorb all the water, endosmosis will happen, but it will not burst. It will reach to its maximum shape or size. So that is mainly because of both vacuoles as well as because of cell wall. It provides turgidity and rigidity. Now, in case of amoeba has two types of vacuoles. The first one is called contractile vacuole, which will help in excretion or osmoregulation. Osmoregulation means regulating the water content. Now, in case of amoeba, again, or some other protist, food vacuole is also there. So food vacuole, like we saw earlier, food vacuole will be mainly involved in engulfing the food particle. So food vacuole along with the lysosome, they will fuse together and helps in endocytosis or phagocytosis. That's all about vacuoles. You don't have to study in detail, only this much is enough for vacuole. Remember that it is membrane-bound organelle. It contains water, sap, excretory products. It is surrounded by a single membrane, and this membrane is called tonoplast. In plant cells, vacuoles are very large in size. Up to 90% of the cell volume will be taken by vacuoles. In plants, the vacuole is called a sap vacuole, which provides turgidity. In amoeba, there are two types of vacuoles, contractile vacuole and food vacuole. Contractile vacuole mainly helps in excretion or osmoregulation. So let's watch two videos. First one, let's watch a video about phagocytosis. So this is an actual real video of how a bacterial cell is destroyed or eaten by our WBCs. So this structure that you see on the screen right now, it is called a phagosome. So phagosome is actually a vesicle containing the bacteria fused with the lysosome. So the enzymes in the lysosome will kind of destroy the bacterial cell. So we'll watch another video. One second. So in this video also you can see here that is a neutrophil, which is a WBC chasing a bacteria. So that big thing is the WBC and that black colored thing is the bacteria. Now it is going to engulf it. There's no sound for this video. So this is actually happening under the microscope. It's a time-lapse video. Okay. Next one, we'll watch a video on cell membrane structure, which we discussed in the last class.
The cytoplasmic membrane of a prokaryotic cell is the main gateway for movement of molecules in and out of the cell. This membrane may also be called the cell membrane or plasma membrane. Cytoplasmic membranes in bacteria are comprised of a phospholipid bilayer. Each phospholipid contains a phosphate head located on the outer part of the bilayer, attached to a glycerol and 2-fatty acid, which face the core of the membrane. The phosphate is negatively charged and hydrophilic, while the tail portion of the phospholipids is hydrophobic. Thus, cytoplasmic membranes are very effective at blocking passage of many molecules, while other molecules are allowed to pass. Cytoplasmic membranes also contain 2 types of proteins. Integral proteins often span the entire bilayer and act as channels for transport of molecules that normally wouldn't be able to penetrate the bilayer. Peripheral proteins are loosely attached to the membrane on one side or the other. Some of these may be glycoproteins, which are proteins bound to a polysaccharide. These proteins may function as recognition sites, enzymes, or receptors.
To Moo Math and Science. In this video, I would like to talk about the Endoplasmic Reticulum, also called the ER. The ER is found in both plants and animals, but not in bacteria cells. If you take a look at the ER, you will notice that it consists of folded membranes and looks like a transportation system. The ER is located near the nucleus and helps the cell transport important materials throughout the cell. Let's look more closely at the function of the endoplasmic reticulum. First, there are 2 types of ER, rough and smooth. Rough ER, or RER, is located very close to the nucleus. The rough ER gets its look because the tiny protein factories called ribosomes are embedded in its membranes. The rough ER produces proteins. Some of these proteins will be transported by the ER to the Golgi Apparatus. Others will move across the ER to other parts of the cell or will be packaged in small sacs which will pinch off from the ER and travel throughout the cell. Smooth ER looks smooth because it does not have any ribosomes. It has a different job than the rough ER. The smooth ER produces important chemicals called enzymes. Some of these enzymes will pinch off in a vesicle and travel to the Golgi apparatus. The smooth ER also produces lipids, which are important fats used by the cell. The smooth ER will transport these lipids throughout the cell or will package the lipids in sacs called vesicles and release them into the cytoplasm. In summary, the ER is a transportation system used by the cell to create and transport proteins, enzymes, and lipids throughout the cell. Thanks for watching. Moo Math and Science uploads a new math, science. Welcome to Moo Math and Science. In this video, I'd like to talk about the Golgi Apparatus. You may have heard the Golgi Apparatus is a bit like a post office. It receives proteins, repackages them, then sends them out into the cell, like a post office does. In fact, that's a little bit what the Golgi Apparatus does. But first, where is the Golgi Apparatus located? It is located by the ER. The side closest to the ER is called the cis Golgi, and the outside is called the trans Golgi. It is found in all eukaryotic cells, so it's found in plant and animal cells. First, the ER repackages proteins and biochemicals into vesicles. It then pinches these vesicles off and sends them to the Golgi apparatus. The Golgi apparatus receives these proteins, lipids, and other chemicals from the rough ER and modifies and packs them into vesicles. These vesicles then pinch off and travel to 3 locations. They may travel to the lysosome and carry digestive enzymes to help the lysosome with digestion. Secondly, some travel outside the cell. They go to the membrane and travel outside the cell, like insulin that will help with the blood sugar level. And finally, some travel to the plasma membrane and provides proteins for repair and replacement. Hope that